What if I told you that we just gave CRISPR, a gene editing tool, the ability to read Google Maps? That scientists at Stanford just invented a way to send healing molecules to specific parts of a nerve cell like DoorDash delivers tacos. And that this could help reverse brain damage from spinal cord injuries or diseases like ALS and spinal muscular atrophy. Let's break down what this spatial RNA medicine actually means and how it could change the future of neurological treatment forever. Inside your neurons, those are your brain's long, branching communication cells. Healing is a team effort. If a neuron gets injured, special messenger molecules called RNA rush in to help fix the damage. Think of RNA as the body's version of a construction crew. Specifically, it's like the foreman who brings the blueprints for how to make the right proteins. But here's the problem: neurons are long, like millimeters to feet long. They're like stretched out spaghetti, and when they're injured, they don't have time for a scavenger hunt. To heal correctly, RNA needs to get to the exact part of the neuron that's damaged, not somewhere nearby. We're talking cellular GPS precision. But in diseases like ALS and spinal muscular atrophy, or in serious injuries like spinal cord trauma, the body's natural delivery system breaks down. RNA ends up getting lost, and when it can't reach the injury site, the neuron can't heal. Damage becomes permanent. Enter the scientist at Stanford, led by Dr. Stanley Chi. You might know him as one of the early architects of CRISPR gene editing, but this time he's done something new. They developed a tool to deliver RNA to a precise location inside a neuron. Not just edit DNA, not just insert RNA. We're talking hyper targeted RNA therapy. They call it spatial RNA medicine. So, how does it work? They created a zip code system using CRISPR's protein tools. Basically, they attached a molecular tag to the RNA, like slapping a GPS pin on a package. And just like how your mail ends up at your house and not at your neighbor's, hopefully, this tag tells the RNA exactly where to go in the cell, down to the specific branch of the neuron. This system is built on a customized version of CRISPR Cas systems, not for cutting DNA, but for ferrying molecules inside the cell with precision. Let's rewind for a sec. What is RNA? DNA is your body's instruction manual, but DNA lives in the cell's nucleus, like a protective library. To get instructions out into the wild, cells make RNA, short for ribonucleic acid. RNA is a copy of the DNA message that can go on a road trip. This RNA travels to different parts of the cell and tells the protein factories, called ribosomes, what to build. If DNA is the architect, RNA is the foreman on site, making sure the blueprints get followed. And the proteins? They're the bricks, wires, and paint. Literally the stuff that builds or fixes the cell. Here's why this is such a big deal. If we can guide RNA to only where it's needed, we don't waste time or resources, and we avoid side effects from scattershot treatments. More importantly, this might actually allow neurons to regrow. That's like saying we could reverse brain damage. In their studies, Stanford's team showed that their zip coded RNA delivery could restore local protein production and support neuron repair. It's not a full treatment for ALS or spinal cord injuries yet, but it's a massive step forward toward targeted regenerative therapies. So let's recap. Neurons need RNA at injury sites to heal. Diseases mess this up, which causes permanent damage. Stanford invented a zip code system using CRISPR. It sends RNA directly to the problem area. This could change the game for neurodegenerative diseases and injuries. This is the foundation of an entire new field, spatial RNA medicine. In the future, we might be able to deliver custom RNA messages to the brain like software updates, telling specific neurons, hey, repair yourself, here's how. It opens the door not just for spinal cord injuries and ALS, but maybe Alzheimer's, Parkinson's, and even aging related decline. Right now, it's still early days, cell and animal models. But it's one of the most elegant solutions we've ever seen for fixing one of the body's most complex systems. And it all started with giving CRISPR a GPS. So the next time someone says CRISPR can't fix the brain, tell them, yeah, well, it can deliver healing instructions to broken neurons now. So, shout out to Dr. Stanley Chi and the Stanford team for redefining precision medicine. If you want more breakthroughs, hit like and subscribe.